in the shoot system of all the land plants, a substance is a type of substance is deposited upon or over the epidermis. That substance is called as cuticle layer. A special layer is deposited upon the epidermal tissue system or upon the epidermis, that is called as cuticle. For example, this is epidermis layer. It is covered by a special layer called as a substance layer called as cuticle. It is made up of a substance called as cutin. This layer is known as cuticle. Okay. Now here cuticle is almost present on all the plants above the epidermis, but in some plants like submerged hydrophytes, and in some roots, you will see the presence of no cuticle. Now, almost all the plants will have cuticle above the epidermis. It is a special layer which acts as a cover, protective cover to the epidermis. Like we have soap box covering the soap. So if soap is a epidermis, then the soap box will be a cover to the epidermis. So that is the function of cuticle. And here it is almost not seen in the roots and submerged hydrophytes. Now, if it is present in the form of, there are two terms here. If there is a continuous formation of cuticle layer, if it is in the form of a continuous layer on the epidermis, then that term is called as cuticularization. If the occurrence or continuation of cuticle is there on the epidermis as a layer, then that is called as cuticularization. If the cuticle is deposited in certain areas on the epidermis, then that is called as cutinization. If there is a deposition of cutin at certain places on the epidermis, then that is cutinization. If the occurrence of cuticle is in the form of a continuous layer, then that is called as cuticularization. Now this cutin is made up of what? What is this cuticle is deposited? For example, we said that cutinization in musa, in banana. Okay, how is the deposition occurrence of cuticle? Or cuticle occurs in the form of certain deposits, and it will develop, these deposits will develop into peg-like structures. We said cutinization. Normally in every plant you will see cuticularization, continuous layer of cuticle above the epidermis. But cutinization, for example, we can give about banana or musa, where the cutin is occurring in the form of deposition, and this deposition ultimately lead to the formation of peg-like structures. This ultimately lead to the formation of peg-like structures. Now this cutin is formed mixed with many fatty substances. Hexadeconic acid. Okay, normally cuticle is formed by the mixture of fatty substances, but in case of angiosperms and gymnosperms, the composition of cuticle is made up is because of the presence of this chemical composition, or because of this chemical, that is 10,16 dihydroxyhexadeconic acid, which is present, because of which the formation of the cuticle occurs. Normally, what is the epidermis cell wall is made up of? The cell wall of the epidermis is made up of, as we all know, it is made up of cellulose and pectin. Okay. Now this cellulose and pectin are the cell wall composition of epidermis. Now where this cuticle is present, then this cuticle is present in association with the cell wall of the epidermal cells, or it is found attached to this cellulose and pectin layer. And here, when this cuticle layer or cutin is reacted with a substance called as ruthenium red, then it will turn into a color called as dark red color. You got my point here? The cell wall of the epidermal cells are made up of cellulose and pectin. So this cuticle layer or cutin which is there inside the cuticle is present, associated very much near to the cellulosic and pectin layer. And how we identify that this is a cutin layer? When this cutin layer, so when they are coming in a close association with cellulose and pectin layer of the epidermal cells, we cannot differentiate between cuticle and epidermis. So how you differentiate? When this particular area is treated with ruthenium red, this area which is having cuticle layer will get converted into dark red. So when we see after treatment with the ruthenium red, we can identify that epidermal cells will have a dark red layer covering on above them. So that dark red layer indicates that that particular epidermal cells are protected by a protective covering called as cuticle. Example for this. Now, in addition to that, we will also see, for example, in the case of pinus needle. We will see the deposition of lignin in the cell wall. Deposition of lignin in the cell wall. Normally we said the cell wall is made up of cellulose and pectin, but in case of pinus needle we can clearly see the presence of lignin deposition on the cell wall. Okay. Now when we come to the thickness of the cuticle layer, when we talk about the thickness, it is not uniform to all the plant. It varies depending upon the species and depending upon the varieties of the plant which we are studying. For example, in case of drought plants, drought area, the plants which are growing in drought areas, the layer thickness is, the cuticle thickness is very thick. The plants which grow in drought areas, the thickness of the cuticle will be much thicker when comparing to the other layers, other plant material. Now what is the use here? Why it is thick here? The thickness of the cuticle will help in the prevention of transpiration in case of, helpful in the prevention of transpiration in case of drought area plants. Okay. Now whereas in case of remaining mesophytes, it will remain as a single layer, as a normal single layer which we can see above the epidermis. Okay, now in case of drought area plant, the plants which grow in drought area, we said the cuticle thickness of the cuticle is more, the layer of the cuticle is much thicker, because which may help in the prevention of transpiration. Whereas in mesophytes, we will see the occurrence of the cuticle is very thin comparing to the drought area plant, and it will be occurring in the form of simple layer. Now when we come to the, for example, this is epidermis with the cuticle. This is epidermis and this is cuticle. Okay. Now when it is an epidermis and cuticle, the epidermal characters are printed on the cuticle. For example, this is out of your syllabus, but what is the major cuticle is there? For example, in fossil plants or something, you get cuticle of particular plant, epidermis is missing or epidermis is ruptured, all that. But though we are able to get all the characteristic features of the epidermis of the some fossil plants. So how it is occurring? Because the epidermal characters of the plant body are printed in the cuticle in the form of an impressions. The impression of all the, as the cuticle is very near to the cell wall of the epidermis, so then these characters of epidermis will be imprinted on the cuticle with the process called as impression. Now when these epidermal peelings are removed and cuticle is available, when we see the cuticle, you will clearly see the arrangement, the composition, every detailed study of your epidermis can be seen in the cuticle also. Okay. Now next, there is one more structure called as, when we talk about cuticle, we are supposed to talk one more aspect of epidermis, that is called as wax.